The overall goal of the following experiment is to determine if a defect generated in the neuromuscular function of C. elegans may be explained by defects in the subcellular compartments of muscle. This is achieved by picking worms from plates and completing movement assays to determine if the mutation, drug, or RNAi treatment has an effect on muscle function. Next, animals containing GFP fusion proteins localized to mitochondria and/or sarcomeres are imaged to identify structural disturbances to mitochondrial networks or the contractile apparatus. Then, the animals were exposed to MitoTracker in order to assess mitochondrial membrane potential in vivo, and beta-galactosidase assays are carried out to determine if cytosolic protein degradation is occurring. The results show whether defects in subcellular compartments of muscle may be responsible for movement defects based on abnormal appearance of mitochondria or sarcomeres, abnormal uptake of dye into mitochondria, and/or abnormal protein degradation.
And these methods can answer key questions about muscle physiology, such as which genes contribute to movement and why.
Though these methods can provide insight into the effects of mutations, drugs, or RNAi treatments, they can also be employed to look at the effects of aging.
Generally, individuals new to this method will struggle with prospective movement assays as worms tend to get stuck in pipette tips.
Demonstrating the procedures will be Joseph Bass and Christopher Gaffney, 2 PhD students, and Thomas Barratt, an undergraduate student from my laboratory.
Handle and maintain C. elegans and construct strains containing CCIS 4251